The overall goal of this procedure is to determine the mobility of enhanced green fluorescence protein by measuring the FRAP of the protein in a region of interest. This is accomplished by first transfecting the EGFP vector in cultured rat hippocampal neurons. Next, FRAP recording is performed in a spine of interest using a Zeiss 710 confocal microscope. The photobleaching rate can then be calculated using ImageJ and GraphPad Prism software. Ultimately, results can be obtained that show the mobility of EGFP in a spine from a cultured hippocampal neuron through confocal microscopy.
This protocol was developed by Chen-Ying Zheng in collaboration with myself, Ronald Petralia, as well as Yaxian Wang, Bashara Kashar, and the senior author of the original paper, Robert Wenthold, who died in October 2009. We dedicate this video to his memory. This method can help answer key questions in the protein dynamics field, such as turnover rate of a protein of interest in a region of interest.
Begin this procedure by culturing embryonic day 18 rat hippocampal neurons on poly-D-lysine-coated MatTek 35mm glass bottom dishes. On 16 to 18 days in vitro, transfect neurons using the CloneTech CalPhos mammalian transfection kit. Before beginning the transfection, save the original culture medium in a sterile 15ml tube for later use. Fill each 35mm dish with 1.5 ml of Delbecco's modified Eagle medium 30 minutes prior to transfection. Next, combine 10 micrograms of pEGFP N1 plasmid DNA with 12.4 microliters of 2 molar calcium solution and bring the total volume up to 100 microliters with sterile water. Add the DNA mixture dropwise to 100 microliters of 2x heaps buffered saline, vortexing the buffer after each drop is added. After letting the final mixture sit at room temperature for 20 minutes, add the buffered DNA solution to the DMEM incubated neurons. Incubate the neurons at 37 degrees Celsius for 1 to 1.5 hours. Following incubation, remove the calcium phosphate containing medium from the culture dishes and wash the cells with DMEM, repeating the DMEM wash a total of 3 times. Exchange the DMEM medium with the original culture medium before returning them to the incubator. Neurons are used for the FRAP experiment 2 to 4 days after transfection. To begin, replace the culture medium in the 35mm glass bottom dish immediately with pre-warmed Tyrode solution. A Zeiss LSM 710 confocal microscope is used for the FRAP experiment. The Zeiss temp module system maintains the temperature, the humidity, and the percent carbon dioxide of the working system. Make sure that the carbon dioxide tank is connected and that the water bottle, which is used for balancing humidity, is filled with water. Once the microscope is set up, find a transfected mature dendrite with the 100x objective. If the transfected cells in the dish are sparse, search for a desired cell with the 40x objective. Then switch to the 100x objective to capture images. Use 5x optical zoom and a 256 by 256 pixel resolution to image a short piece of dendrite with several spines. To capture images, use nominal speed 9, which takes 0.5 seconds to finish a scan. Set the pinhole to 2 micrometers to obtain strong fluorescence. When taking images, try to use low laser transmission, for example 1 to 5%, to avoid photobleaching the entire image. Next, select the spine of interest. For this experiment, mushroom spines with spine head diameters of about 1 micrometer are chosen. To perform a FRAP experiment, take 5 control images before bleaching. Then bleach the spine of interest 10 times at nominal 100% laser transmission. Capture a series of images immediately after bleaching. The interval of time should be adjusted according to different targeting proteins and different experimental designs. For this experiment, images are captured every second for 15 seconds after bleaching. Next, save the images. To analyze the data, open images with ImageJ software. Align the stack of images using the Align tool so that the spine of interest does not float and remains in the same position on the image. Measure relative fluorescence intensity of the spine of interest, which is a transfected but unbleached region. Measure the background region in time-lapse images with the intensity versus time monitor tool of image J. Use a non-fluorescent region as the background region. Curve fit the fluorescence intensity with a one-phase exponential equation in GraphPad Prism software or other similar software. Finally, calculate the % mobile and immobile fraction of fluorescence. Shown here are the FRAP measurements of EGFP fluorescence in a spine from a cultured hippocampal neuron. The region of the spine, control, and background are marked, and the red arrowheads indicate the time of photobleaching. Photographs of the same area were taken before and at 0, 1, 5, 10, and 15 seconds after photobleaching. FRAP curves of EGFP fluorescence are shown over a 15-second period, with the dots on the curves representing the FRAP at every second. The curves were fitted by one-phase exponential equations. The average fluorescence before photobleaching was counted as 100%. In this experiment, the mobile fraction is 87% and the immobile fraction is 13%.
After watching this video, you should have a good understanding of how to measure the mobility of a GFP-tagged protein by using FRAP technique.